committee to invite me to give a lecture. Today I will talk about the drug discovery program, how it runs in our lab, and in particular about the novel non-RGD cyclic peptide drug conjugates for targeted treatment of human metastatic melanoma. Before I start, just a short overview of targeted drug delivery methodologies that are currently in use. First of all, First of all, the most known is ADC, the antibody drug conjugates, NDC, nanoparticle drug conjugates, PDC, peptide drug conjugates, the other methods like polymer drug conjugate, protein drug conjugate, polymeric micelles, and so on, so on. We are here. We are working with peptide drug conjugates. We have reason for that. First of all, the peptide drug conjugates are not immunogenic. They are completely synthetically reproducible. They are applicable for high throughput synthesis because we do it by solid phase chemistry. And are also applicable in student lab, not less important. So how we do it? First of all, we choose the peptide carrier. Then we link amino acid platform, specially developed. So this amino acid platform will link small molecules through the biodegradable linkage. And then this conjugate is ready for targeted drug delivery. We can use, instead of peptide carrier antibody, the chemistry will be different, or nanoparticle as well. But we prefer to work with the peptide carrier from the reasons I mentioned. The small molecules can be any small molecule that possess a linkable hook, linkable moiety, for creation of those biodegradable moieties. We work with something like 15, 20 small molecules. Here is an anti-cancer project. And small molecules are, here is a chlorambucil, all DNA alkylator with the carboxylic group, a 6-MP with a thiol group, antimetabolite. This one, amonophyte, they failed in phase 3. Topo 2 isomerase inhibitor and frequently appeared, used in many clinical trials. This one is camptothecin having the phenolic tether. Compretostatin having the phenolic tether, which is an antimitotic agent. And this one is very old, topo 2 isomerase inhibitor, AHMA. So for using small molecules in this model, we need hooks that through these moieties, we can link them to the amino acid platform. About the linkage strategy. Basically, the linkage strategy is divided in two. Those that are protease sensitive, enzymatically sensitive, or sensitive in general, and those that are not sensitive, not cleavable. In cleavable linkers, the most known are protease sensitive, for instance, valine, citrulline here. Can be a pH sensitive, like hydrazone here, and also this will fit here, which is glutathione sensitive. We prefer to work with the traditional linkers, amides, phosphate esters, carbonite, in the same matter as they appeared in prodrugs. Let me show you the diversity of our peptidyl-based platform. This is model of 3 drugs linked through the amino acid platform, consists of 2 amino acids, which is linked to the peptide carrier. All the synthesis from the beginning to the end is done by solid phase chemistry, applicable for high throughput synthesis. Amino acid platform can be any amino acid platform which carries appropriate tether, like a phenolic tether, carboxylic tether, and so on and so on. Serine, threonine, aspartic acid, ornithine with the amine tether, cysteine with thiolic tether, unnatural amino acid, anything that get in here and create the biodegradable moieties here. And also, the chirality matters. So what we can say that our platform is highly tunable. And this being tunable is extremely important because we have to catch very narrow therapeutic window in order to release the drugs inside the target, inside the cancer cell, not outside. So by tuning, having the capabilities of tuning this release profile, we can gain that. So basically, if I would summarize what I said, we aim to targetedly deliver chemotherapeutic cocktails to reduce toxicity and increase efficacy. Anybody who is dealing with targeted drug delivery do the same to reduce toxicity and increase efficacy, but we do it to the cocktails. I can show you, and we gain the reversing of the drug resistance by targeted drug delivery. Drugs that cells that have been resistant to certain small molecules became sensitive when drugs are linked to the carriers. And we prefer to use all drugs or failed in clinical trials candidates. Why? Because the contrast is immediate. I mean, something which is drugs that are failed because of toxicity, when they are linked to the peptide carriers, they are less toxic and even more efficient. The pharmaceutical garbage is huge, so it's so many molecules inside that reach phase 3. So many money been waste in vain. We think it's a good stuff for reusing. So I would like to show you 2 projects. The first one is sort of model. Another one is a real one. The first one, we took alpha V beta 3 integrin system. We used the silengtide, which is an advanced analog of alpha V beta 3 integrin inhibitor. It's failed as far as I know in phase 3 because of lack of efficiency, but it's a very good model because it's very potent to those integrin subtypes. And we prepared monoconjugates. This one is the peptide with the chlorambutyl through the amide bond. This one chlorambutyl through the ester bond. We substituted the lysine with the serine. And here is camptothecin, topo 1, very powerful isomerase inhibitor, very toxic, but not bioavailable, conjugated through the amide bond. And at the same time, we prepared big conjugates. Here you can see the same peptide with the 2 chlorambutyl. And the first molecule, 2 molecules are linked through the amide bond, and the second one through the amide, another through the serine. Here are 2 molecules of camptothecin through the carbamide bond, and here is a real cocktail. I mean, heterogenic. It's a camptothecin topo 1 isomerase inhibitor, and the second one is a DNA alkylator chlorambutyl. This is camptothecin, this is chlorambutyl. So basically what we try to do, why we want to use cocktail, because we hope that by shutting down in parallel several oncogenic mechanisms, we can gain better efficacy. And results are here. So we took a non-small cell lung carcinoma cell line. This is murine melanoma. And this is HEC. Those 2 are overexpressed almost 90% with alpha V beta 3 integrin. This one less than 3%. We checked by Fox analysis. And this monoconjugate was compared to the big conjugate. So we wanted to check if the higher accumulation of the drug inside the cancer cells will be more effective. So according to results, you can see here, those 2 cells that are overexpressed with alpha V beta 3 integrin are sensitive to camptothecin and monoconjugates and also big conjugates in the dose-dependent matter. The most effective was big conjugate here. RGD itself is not active. Here in HEC, camptothecin is the only molecule who is sensitive, who is effective in dose-dependent manner. The P conjugate is not working, but see the difference here, B conjugate versus here and here. So this was a good model for proving us that conjugation of several molecules to the same carrier can result in better efficacy. Then we took heterogenic conjugate, and compared to the monoconjugate. The first molecule conjugate was bearing camptothecin, and this is chlorambucil. And this one is a binuclear conjugate bearing chlorambucil and camptothecin on one peptide. Results are here. The same model. Overexpressed more than 90% non-small cell carcinoma, overexpressed more than 90% non-small cell marine melanoma, and this is HEC, very low expression. So, as you can see, the monoconjugates were less effective than heterogeneous B conjugate, and here the B conjugate, which was very powerful, very effective here, is not effective here at all, while the camptothecin in the free form was toxic to HEC, toxic to non-small cell lung carcinoma, and to the cell line, cancerous cell line. So being encouraged, I forgot the arrows. We moved to another project, which was bigger, and decided to develop our own peptide carriers for this system, alpha V beta 3. We applied a flash display methodology and came out with this interesting peptide, small peptide, cyclic peptide, you see here. And this peptide eventually appeared to be non-RGD, alpha beta 3 integrin binder. That was by chance, but it's very important because there have been reported several times that RGD peptides can cause some cell proliferation that can lead to the secondary cancer. So having non-RGD carrier is very interesting to investigate. We did some docking simulations, and we saw that our peptide binds here while the RGD site is here. So, and we did some comparative FACS analysis, and we're quite confident that our ALOS4 peptide binds to the different from the RGD site. This ALOS4 appeared to be very interesting and promising anti-cancer peptide. You can see here the lung of the mice. The difference between them is that this one, the model is murine melanoma. So those are metastases. This lung is from animal that was not administrated with ALOS4, and this one that was administrated with ALOS4. Here you can see the mortality was reduced by administrating different dogs. Don't want to heal mice, want to heal human, right? So here we switch to the metastatic melanoma cell line, this one WM2664, and tested the accumulation, the selective accumulation of the ALOS4 fits in cancer cells. So this is fits, and this is ALOS4 fits. As you can see, very nice accumulation inside the cancer cells. So we did some in vitro toxicity. We took HEC versus metastatic melanoma. This one, more than 90% overexpression of alpha V beta 3 integrin receptor. This is less than 3%. It's a good model. We took a free azotoxin. This is also topo-2 isomerase inhibitor, which was failed in clinical trials. We like molecules that are failed in clinical trials very much. Finito. Okay. No battery. Works, sorry. All those three molecules were compared to this conjugate, which is allos 4 linked through the carbamide bond to the camptothecin. Free camptothecin, free ammonophyte, free azotoxin versus a camptothecin conjugate. And results are as follows. Okay. Oh, yeah, we can see. In HEC, the free drugs, the most toxic was camptothecin. Allos 4, camptothecin was not toxic at all. No alpha V beta 3 overexpression. Cannot penetrate. Here on the metastatic melanoma, you can see that allos 4, I can't see, but you probably can see. You can see it from here, that the allos 4 linked to the camptothecin was the most effective and most toxic, while the free camptothecin was not toxic. So it's sort of reversing the resistance. So those cells are resistant to camptothecin. Now by linkage to the peptide carrier, they are sensitive. The recent confocal microscopy result, just to show you that we know that after 1 hour, they're accumulated in lysosomes, and now we're in the middle of the study for time-dependent research. What happens to this peptide linked to several drugs inside the cell, distribution inside the organelles, and so on, because it's important to be focused on the mechanistic issue of this conjugate and others too. Then we did ex vivo fluorescence imaging on the same cell line. So we developed an animal model with a human metastatic melanoma, and ex vivo showed clear accumulation of allos 4 fits only in tumor, while the liver and spleen are clean. So it's a good point to move forward. The Western blot also was interesting. So we compared HEC, low expression again of alpha V beta 3, with the metastatic melanoma, high expression of the alpha V beta 3 integrin receptor. We took 2 kits. The first one was caspase 3, responsible for apoptosis. Another one was H2A XP, responsible for DNA fragmentation. Okay. So as you can see here, after 24 hours, only the camptothecin in the free form in HEC cell line causes the apoptosis. Allos 4 is not active. It cannot enter the cancer cell. Here you can see that camptothecin causes apoptosis and DNA fragmentation, but also in the same matter our conjugate during this camptothecin. But this conjugate is not toxic here. So what is also interesting to see that after 3 hours, allos 4 here is not working. That means that the drug was not released from the peptide after 3 hours. It probably should wait more. So now we're investigating the time, the release regime of this drug from the peptide by using the Western blot. That's it, 20 minutes.
Thank you very much.
No, but conclusion, just a moment.
Conclusion.
So we developed the high-throughput loading methods for cocktails onto biodegradable peptide platforms. We established biostability and chemostability profiling. This is extremely important. We do a huge work in different pHs, different media, and liver homogeneate, the mice liver, not human, yeah? Mice liver homogeneate, different lizards, whatever, because this release profiling is extremely important for optimizations process of these conjugates. In order, as I said, to tune it properly for getting inside very narrow therapeutic window. We don't want all this conjugate to travel around the body and release this payload in different places that we don't want there, only in the cancer, preferably. We have shown increased targeted toxicity of peptide drug cocktail conjugates versus peptide monodrug conjugates. So the higher accumulation of those drugs inside the cancer cells is more effective. We developed non-RGD alpha V beta 2 integrin specific peptide carrier ALOS4 for targeted drug delivery in human metastatic melanoma. Currently, we are working on the Vivo study on other solid tumors. And I would like to thank the team. The chemistry was done by mostly by my lab. Some of PhD students are sitting in this hall. Biology was done by Professor Albert Prihasov. The ALOS4 was developed in his lab with my associate Michael Fear and so on and so on. Modeling was done by Dr. Dortobi. We have some grants, and thank you for your kind attention.
Thank you. Thank you very much. Your timing was perfect, 20 minutes. So the noise you heard came from the background, it wasn't us.
Okay.
Okay, so I don't know what's going on. So please, 1 or 2 questions. Just wait.
I've seen in your conjugates, you have probably esters, amides, and carbamates. Those are the 3 main ones. So carbamates and esters would probably cleave more easily, but how do you cleave the amides?
It's a very interesting question. The amides do have some sensitivity. When we did the chemo and biostability, it cleaves, but much more difficult than ester and carbamates. But sometimes this conjugate works without cleaving, without cleaving the drugs as it. This particular molecule bears 2 DNA alkylator, mustard alkylator, very active. So it works, but we don't know inside the cancer cell by release from the carrier or by as conjugate itself. You are right, the amide may be not so good to use, but it works. We don't know why. Probably for the future, we better use esters, carbamates, phosphonic esters, carbonate. You are right, but for model, amide is a chemical moiety. What I can do.
Any other question? You mentioned that you are going to study the timing of
Yes.
release of drugs.
Yes.
How you are going to do this to control the timing?
Oh, first of all, using the confocal microscopy, I think we take it out time dependently, fix it is the first way, and see. Another way on the living cells. So we have a camera that can just take a shots every half an hour or whatever in different intervals of time. For the Western blot, which is the stop it, the process and see what's happening there according to the markers. I think it's a lot of work, but I think it works. And then we want to go to the DNA chips in order to see the genes, what the genes are involved inside. And altogether, hopefully we'll come out with some idea about the mechanism. Because it's not necessarily the same mechanism as a free drug. We don't know. We have to compare.